Think back to the last time you were sitting in a quiet room, perhaps late in the afternoon, and you felt that unmistakable, heavy pull behind your eyes. It isn't a choice you make. It's a physical weight, a gravity of the mind that seems to tug your eyelids downward. You might have fought it, reaching for a cup of coffee or splashing cold water on your face, but eventually, that weight always wins. Why is that? Why is it that the most brilliant machine in the known universe, your brain, requires us to spend a third of our lives in a state that looks, from the outside, like total vulnerability? Welcome to this quiet space of learning. Tonight, we aren't just looking for rest. We are looking for understanding. We are embarking on a 15-lesson journey into the very chemistry of your consciousness. If you find yourself drifting off as I speak, do not fight it. Your brain is designed to absorb, to integrate, and to learn, even as you surrender to the night. In fact, that is exactly what we are here to explore. Before we begin this deep dive into the molecular ballet of your mind, I invite you to subscribe and stay with us through all 15 chapters. By the time we reach the end, you won't just be waking up. You'll be waking up with a profound appreciation for the invisible work your brain does while you dream. Let's begin where all journeys in biochemistry begin, not with a formula, but with a question. Lesson 1. Foundations of Sleep, Neurobiology. For centuries, humanity viewed sleep as a lapse, a blank space in the ledger of our lives. We thought of it as a passive state, a simple turning off of the lights. Even early scientists believed the brain essentially shut down to save energy, much like you might power down a computer at the end of a long workday. But as our tools for peering into the skull became more refined, we discovered something startling. Your brain is not a computer that powers down. It is more like a city that never truly sleeps, but rather one that changes its entire economy and infrastructure once the sun goes down. To understand the neurobiology of sleep, we must first look at the brain as a garden. During the day, the garden is a place of frantic, beautiful activity. The sun is out, photosynthesis is occurring, insects are buzzing, and the plants are stretching toward the light. This is your waking state. You are consuming information, reacting to stress, and burning through energy. But a garden cannot sustain that level of growth indefinitely. If it did, it would eventually choke on its own success. The soil would be depleted, and the paths would be overgrown with weeds. Sleep is the time when the gardener comes out under the moonlight. This gardener doesn't just let the plants sit in the dark. They are active. They are pruning the branches that grew too far, watering the roots that are thirsty, and most importantly, they are clearing away the debris, the fallen leaves, and spent blossoms that would otherwise rot and stifle new growth. When we talk about neurochemistry, we are talking about the tools this gardener uses. We are talking about the specific molecules that signal when it is time to stop growing and start repairing. In the academic world, we describe the foundation of sleep through something called the two-process model. This was first proposed by a researcher named Alexander Borbély in the early 1980s, and it remains the bedrock of how we understand why we feel sleepy. Imagine two different forces working in tandem, like two separate currents in a river that eventually merge. The first force is what we call process S, or the homeostatic sleep drive. Homeostasis is just a grand word for balance. Think of process S as a chemical debt. From the very second you wake up in the morning, your brain begins to accumulate a specific molecule called adenosine. The longer you stay awake, the more adenosine builds up in your system. It is the weight I mentioned earlier, the physical pressure of sleep. If you stay awake for 20 hours, your sleep debt is high. and your brain will demand payment. This is why, no matter how much willpower you think you have, the biochemistry of process S will eventually force your systems to stall. The second force is process C, or the circadian rhythm. While process S is about how long you've been awake, process C is about the time of day. This is your internal biological clock, governed by a tiny cluster of cells in your brain that responds to light and dark. These two processes work together in a beautiful, overlapping wave. During the day, your circadian rhythm sends out alerting signals that counteract the rising pressure of adenosine, keeping you upright and focused. But as night falls, the alerting signals of process C fade away, just as the pressure of process S reaches its peak. It is at this intersection, the meeting of the debt and the clock, that we finally fall into the embrace of sleep. Now, why does this matter to you, sitting here right now? Because understanding this foundation changes how you view your own fatigue. It moves sleep from being a weakness to being a vital chemical necessity. When you feel tired, it isn't your body failing you. It is your neurochemistry successfully signaling that the cleanup crew is ready to begin. A key figure in this foundational understanding was Dr. Nathaniel Kleitman, often called the father of modern sleep research. In the 1930s, he famously spent 32 days living in the total darkness of Mammoth Cave in Kentucky to see if his body would still maintain a sleep-wake cycle without the sun. What he found confirmed that our brains have an inherent chemical rhythm that exists independently of the outside world. We are biologically wired to rest. But what exactly is the brain doing during this rest? We must define another core term: neuroplasticity. This is the brain's ability to reorganize itself by forming new neural connections. During the day, you are creating raw data. You are meeting people, learning skills, and experiencing emotions. These are like loose bricks delivered to a construction site. If you just kept piling up bricks all day, every day, you would eventually have a mountain of rubble, not a house. Sleep is the architect and the mason. It takes those loose bricks of information and cements them into the existing structure of your memory. It discards the bricks that are broken or unneeded and reinforces the ones that are vital. This is why a student who sleeps after a long study session will actually know the material better the next morning than one who stayed up all night, cramming. The chemistry of sleep is the glue of knowledge. Let's look at a real-world example of what happens when this foundation is cracked. Consider the brain fog you feel after only 4 hours of sleep. In that state, your adenosine levels, process S, are still high because they weren't fully cleared out, and your circadian rhythm, process C, is trying to send alerting signals to a system that is still clogged with metabolic waste. It's like trying to run a marathon in a suit of armor while carrying a heavy backpack. Your neurochemistry is noisy. The signals can't get through. This brings us to the concept of consciousness as a spectrum. We often think of awake and asleep as a light switch either on or off. But neurochemically, it is much more like a dimmer switch. There are varying levels of arousal and varying levels of sedation, all controlled by a cocktail of chemicals. In this first lesson, I want you to sit with the idea that your brain is a highly sensitive chemical laboratory. Every thought you have uses energy, and every bit of energy used creates a byproduct that must be managed. As we progress through this course, we will meet the specific molecules that manage this lab. We will talk about GABA, the brain's natural sedative that quietens the noise. We will talk about melatonin, the hormone that acts as the sunset for your internal world. We will explore Orexin, the chemical glue that keeps you awake, and what happens when it goes missing. But for now, just breathe into the realization that you don't have to do anything to make this work. Your brain knows how to take care of itself. It has been perfecting this chemical transition for millions of years. Every animal, from the tiniest fruit fly to the great blue whale, partakes in this foundational rhythm. We are all bound by the same molecular requirements. In our journey together, we will move from these broad foundations into the microscopic details. We will look at how light enters your eyes and travels to a specific spot in your brain to start a countdown. We will look at how caffeine acts like a mask for your sleep debt and why that mask eventually slips. We will even explore the strange, high-voltage world of our dreams, where the brain is more active than it is during a difficult math test. But all of that complexity rests on what we have discussed today, the simple, profound truth that sleep is a restorative chemical process. It is the gardener tending to the mind. It is the clearing of the debt so that you can start the next day with a clean ledger. When you think of your brain tonight, don't think of it as a machine that is breaking down or needing a recharge. Think of it as a vibrant ecosystem that is simply changing its focus. It is moving from the external world of doing to the internal world of being and becoming. You are not losing time when you sleep. You are gaining the mental clarity and structural integrity that allows you to be who you are when you are awake. And with that understanding quietly in place, we're naturally led to ask, what exactly is it that builds up inside us all day, making that pull toward the pillow feel so inevitable? To answer that, we must look closer at the hourglass molecule itself. Lesson 2. The chemical architecture, adenosine and sleep pressure. In our last quiet session together, we ended with the idea of a buildup that signals the need for rest. This is what scientists call sleep pressure, and its primary architect is a molecule called adenosine. To understand how your brain begins its journey toward the night, we have to look at the very currency of life itself. Every moment you are awake, every thought you process, and every movement you make requires energy. In the biological world, that energy comes from a molecule called ATP, or adenosine triphosphate. You can think of ATP as a fully charged battery that powers every cell in your body. When a cell needs to do work, it breaks a piece off that battery to release a spark of energy. What remains after that energy is spent is the molecular scrap, the byproduct of your existence. And that scrap is adenosine. Imagine, if you will, an internal hourglass sitting deep within the structures of your brain, particularly in an area called the basal forebrain. From the very second you open your eyes in the morning, the top bulb of that hourglass is full, and the grains begin to fall. Each grain is a molecule of adenosine, a tiny chemical reminder of the energy you have used. As your day progresses, as you solve problems at work, laugh with friends, or simply navigate the physical world, the bottom of that hourglass fills higher and higher. This is the chemical architecture of your tiredness. It isn't a vague feeling. It is a physical accumulation of metabolic debt. The more you do, the more debt you accrue, and the more adenosine begins to flood the spaces between your neurons. This accumulation is the foundation of what we call Process S, the homeostatic sleep drive. It is a beautiful, self-regulating system. The very act of being awake and active creates the chemical requirement for sleep. It is as if the brain has a built-in accountant, meticulously tracking every expenditure of energy and ensuring that, eventually, you must pay back what you have borrowed through rest. Adenosine is that accountant's ledger. It is the brain's way of keeping score, ensuring that we do not push our cellular machinery past the point of repair. But how does a simple molecule make us feel the weight of sleep? To understand this, we have to look at the receptors, the locks on the surface of your neurons that wait for the adenosine key. There are several types of these receptors, but the most important for our rest are the A1 and A2A receptors. When adenosine levels are low, your brain's arousal systems, the parts of you that are alert and focused, run at full capacity. But as the adenosine builds up, it begins to bind to these A1 receptors. When the key turns in that lock, it doesn't activate the neuron. Instead, it whispers to it to slow down. It inhibits the release of excitatory neurotransmitters like dopamine and acetylcholine. It is like a gentle hand being placed on a spinning wheel, gradually applying friction until the motion becomes a slow, rhythmic crawl. At the same time, adenosine binds to the A2A receptors, which have the opposite effect on the parts of the brain that promote sleep. While the A1 receptors are busy silencing the awake centers, the A2A receptors are busy waking up the sleep centers. It is a perfectly coordinated pincer maneuver. Your brain is simultaneously turning down the volume of the day and turning up the silence of the night. This dual action is why, as the hours pass, it becomes increasingly difficult to maintain high-level focus. Your working memory begins to stutter, your reaction times slow, and your thoughts may begin to drift. This isn't a failure of your will. It is the chemical architecture of your brain, ensuring that you prioritize the recovery that is soon to come. Historically, we have learned a great deal about this system by studying its most famous adversary, caffeine. Almost everyone has experienced the magic of a morning cup of coffee or tea, but few realize that caffeine doesn't actually give you energy. It is, quite literally, a molecular mask. If you were to look at a molecule of caffeine and a molecule of adenosine side by side, you would see a striking resemblance. They are shaped so similarly that caffeine can slide into those A1 and A2A receptors just as easily as adenosine can. Imagine our hourglass again, but this time, someone has placed a tiny piece of clear tape over the hole where the grains fall. The grains are still there, building up in the top bulb, but the sensor at the bottom can't see them. Caffeine is that piece of tape. It sits in the lock of the receptor, but it doesn't turn it. It is a competitive inhibitor. Because the caffeine is taking up all the parking spaces, the adenosine has nowhere to go. It continues to circulate in your brain, building up to higher and higher levels, but you don't feel it. You feel alert, perhaps even jittery, because the slowdown signal is being blocked. However, the laws of biology cannot be cheated forever. Caffeine eventually breaks down and is cleared by your liver. When that happens, that piece of tape is removed, and all the adenosine that has been accumulating behind the dam for hours suddenly rushes in and floods the receptors all at once. This is the caffeine crash. We know all too well. It is the moment the accountant finally catches up with you and demands the full payment of the day's debt with interest. This real-world connection helps us see that adenosine isn't just a byproduct. It is a vital signal that we ignore at our own peril. We can also see the elegance of this system in the phenomenon of the power nap. You may have noticed that even a 20-minute sleep in the middle of the day can make you feel remarkably refreshed. This is because even a brief period of sleep allows the brain to begin the process of clearing adenosine, while it takes a full night of deep, slow-wave sleep to completely empty the hourglass. Even a few minutes of rest can sweep away enough of the molecular scrap to lower the pressure and give you a few more hours of clarity. It is like emptying an overflowing trash bin, just enough to fit a few more things inside. As we look deeper into the layer of this chemistry, we find the work of researchers like Dr. Robert McCarley, who spent decades mapping how adenosine levels rise specifically in the basal forebrain. His work showed that this isn't just a general brain-wide phenomenon, but a localized trigger that tells the executive parts of our brain, the parts that make decisions and plan for the future, that it is time to yield. This serves a profound evolutionary purpose. If we didn't have a chemical way to force rest, we would likely work our hearts and brains into a state of permanent exhaustion. The adenosine system is a protective mechanism, a fail-safe that preserves the integrity of our biological hardware. Think of the adenosine in your system right now as a warm, heavy fog. It has been rolling in all day, slowly obscuring the sharp edges of the world. It is the pressure that makes your pillow feel like the most welcoming place on earth. In the university of your own body, this is your first and most persistent lesson, that every action has a cost, and every cost must be balanced by rest. We are not designed for perpetual motion. We are designed for a rhythmic oscillation between the fire of the day and the cool, chemical waters of the night. When we consider the architecture of this molecule, we see the wisdom in its design. It doesn't use a complex, external signal to tell us to sleep. It uses the very remnants of our own energy. It is the most honest system imaginable. Your tiredness is the direct result of your life. The more intensely you live, the more deeply you will eventually need to sleep. It is a fair trade, a biological contract that we sign every morning when we wake up. As you lie here, you might feel that pressure now, the subtle binding of adenosine to those A1 receptors, the quiet muting of your internal dialogue, the heavy, comfortable sensation in your limbs. This is your brain doing exactly what it was meant to do. It is preparing the ground. It is clearing the way for the other players in our symphony to take the stage. Because while adenosine provides the urge to sleep, it cannot complete the journey alone. It needs a signal, a herald, to tell the rest of the body that the environment is ready for the transition. Let's set that idea of the chemical hourglass down now, fully formed. Let your attention drift away from the buildup of the day and toward the signal that comes from the darkness itself. So what does this structure, this heavy, built-up pressure, actually do once the hourglass is full and the light begins to fade? That's the question we'll gently explore next, as we meet the messenger of the night. Lesson 3. Melatonin, the messenger of darkness. As that adenosine pressure reaches its peak, creating that heavy, undeniable urge to close your eyes, another player enters the stage to signal that the environment is ready for us. This is melatonin, a molecule so tied to the cycles of our world that it is often called the messenger of darkness, or the vampire hormone, because it only truly comes to life when the lights go out. If we think back to our previous lesson, we established that adenosine is the urge to sleep, the buildup of a physical debt that must be paid. But the urge to sleep is not the same thing as the act of sleeping. You have likely felt this yourself. You may be exhausted. Your sleep pressure may be at an all-time high. Yet if you are in a bright, loud, or stressful environment, sleep remains elusive. This is because the brain requires more than just pressure. It requires a signal. I want you to imagine a grand, ancient theater. The audience has arrived and is settled in their seats. This is the adenosine pressure we've been carrying all day. The actors are backstage, costumed and ready to perform. These are the various stages of sleep we will eventually explore. However, despite the audience being ready and the actors being present, the play cannot begin until the lights in the house go down and the conductor raises their baton. Melatonin is that conductor. It doesn't make the audience sit down, and it doesn't perform the play itself. Instead, it provides the official announcement to the entire body that the sun has set, the world has quieted, and it is now chemically appropriate to begin the nightly rituals of repair. To understand where this messenger comes from, we have to look deep into the center of the brain, to a tiny, pine-cone-shaped structure called the pineal gland. For centuries, this gland was a mystery. The philosopher René Descartes famously called it the seat of the soul, believing it was the place where our thoughts and our physical bodies met. While modern science has a more biological view, the pineal gland remains a remarkable organ. It acts as a biological transducer, taking an external signal to the absence of light and translating it into an internal chemical signal, the release of melatonin. The chemistry of this transformation is a beautiful example of the brain's efficiency. Melatonin is synthesized from another neurotransmitter you may recognize, serotonin. During the day, serotonin is often associated with mood, stability, and wakefulness. But as the light begins to fade, the brain begins an elegant alchemical process, converting that daytime serotonin into nighttime melatonin. It is a reminder that our internal chemistry is never static. It is a flowing river, constantly changing its composition to match the world around us. This conversion process is triggered by a lack of blue light hitting the retina of your eyes. When your eyes perceive darkness, they send a message through a specific neural pathway to the pineal gland, essentially whispering, "The sun has gone. You may begin." The history of our understanding of melatonin is relatively recent. It was only in 1958 that a dermatologist named Aaron Lerner isolated the hormone, originally hoping it might be a key to treating skin conditions. What he discovered instead was the master key to our biological timing. Since then, we have learned that melatonin does much more than just signal sleep. It is a powerful antioxidant, perhaps one of the most potent in the human body. As it circulates through your bloodstream, it acts like a systemic, all-clear signal, telling your cells to slow down their metabolism, lower your core body temperature, and begin the process of DNA repair. It is the molecular equivalent of a citywide curfew that allows the maintenance crews to safely work on the streets. This brings us to a very modern dilemma and a real-world connection that many of us feel every day. Because the production of melatonin is so strictly controlled by light, our modern environment has created a state of chemical confusion. In the natural world, the only source of light after sunset was the moon, or the warm orange glow of a fire. Neither of these contain much blue light, which is the specific frequency that tells the brain the sun is still up. Today, however, we surround ourselves with digital suns, our phones, tablets, and LED lights. These devices emit a high concentration of blue light, which tricks the pineal gland into thinking it is still midday. When you look at a bright screen late at night, you are essentially shouting at your conductor to stay backstage. Your adenosine pressure might be screaming for rest, but the melatonin signal is suppressed, leading to that tired but wired feeling. This is why sleep experts suggest digital sunsets or the use of amber-tinted glasses to allow the melatonin surge, what scientists call the dim light melatonin onset, DLMO, to happen naturally. When the melatonin surge finally begins, usually about 2 hours before your typical bedtime, it starts a cascade of physiological changes. Your blood vessels in your hands and feet begin to dilate to release heat, lowering your core temperature. Your heart rate slows. Your breathing becomes more rhythmic. It is a systemic surrender. We see this same mechanism in almost all vertebrates. Even in the depths of the ocean, where sunlight barely reaches, creatures use melatonin to sync their bodies with the rhythms of the earth. It is an ancient, universal language of rest. We also see the importance of this messenger when we travel across time zones. Jet lag is, at its core, a melatonin mismatch. Your adenosine hourglass might be full, but your pineal gland is still synced to the light of a city thousands of miles away. It takes time for the conductor to learn the new theater's schedule. This is why many people use supplemental melatonin to help nudge their internal clock. Though it's important to remember that in this context, it isn't acting as a sedative, but as a chronobiotic, a tool to shift time. In the context of our wise professor perspective, I want you to consider the profound patience of this system. Your brain doesn't just drop you into sleep. It prepares you. It waits for the right environment. It gathers its precursors, and it sends out its herald. Melatonin is a grace note in the symphony of our biology. It is the evidence that we are not separate from the planet we live on. We are deeply, chemically intertwined with its rotation and its shadows. As you lie here, the melatonin is likely performing its quiet work within you right now. It is bathing your neurons, signaling to your muscles that they may loosen their tension, and telling your heart that it may beat a little more slowly. It is the official announcement that the world of the day is over and the world of the night has begun. You are currently floating in the transition that this messenger has facilitated. But as we acknowledge the beauty of this signal, we must also ask, who tells the melatonin when to start? If the pineal gland is the conductor, who is the one holding the sheet music? Who is tracking the 2-4-hour cycle of the earth and ensuring that the melatonin surge happens at the exact right moment, night after night, even if we were to spend our lives in a cave? Let's set that idea of the messenger of darkness down now, fully formed, and let our attention drift toward the master clock that sits just above the roof of your mouth, governing the very rhythm of your life. Before we rest here, let me whisper where we're headed next, not to rush, but to plant a seed. While the melatonin manages the announcement, there is a central command center that manages the entire 2-4-hour schedule. So what does this structure actually do to keep us in sync with the stars? That's the question we'll gently explore next. Lesson 4. Circadian Rhythms. The Suprachiasmatic Nucleus. In our last quiet session together, we ended with the idea of melatonin acting as a conductor. But as we discussed, even the most brilliant conductor needs a score to follow a master script that tells them exactly when to raise the baton and when to call for silence. That score is not written in notes, but in time itself. And the keeper of that time is a tiny, formidable structure known as the Suprachiasmatic Nucleus, or the SCN. To find this master clock, we have to travel deep into the brain, past the surface-level thoughts and the bustling centers of emotion, down into the hypothalamus. This area is the brain's maintenance closet, responsible for all the things you don't have to think about: your hunger, your thirst, and your body temperature. Within this space, Sitting right above the point where your optic nerves from each eye cross over a spot called the optic chiasm, lies the SCN. It is a cluster of approximately 20,000 neurons, which sounds like a lot, but in the vast landscape of the brain's 86 billion neurons, it is a mere speck, no larger than a grain of rice. Yet, this tiny grain governs the rhythm of every single cell in your body. Think of the SCN as the master clock in a grand ancient town square. In the old days, before every person carried a digital watch in their pocket, the town clock was the absolute authority. When it chimed, the baker knew to open the shop, the workers knew to head to the fields, and the watchman knew to light the lamps. Your body is a collection of trillions of local clocks. Every organ, your heart, your liver, your lungs, and even every individual cell has its own internal timing mechanism. But left to their own devices, these local clocks might begin to drift. Your liver might think it's noon while your heart thinks it's midnight. The SCN is the master signal that keeps them all in perfect, rhythmic harmony. It sends out a chemical and electrical chime that ensures every part of you is working on the same schedule. This 2-4-hour cycle is what we call a circadian rhythm. The word circadian comes from the Latin circa, meaning around, and diem, meaning day. It is our internal, around-a-day cycle. But how does this tiny cluster of cells know what time it is in the outside world? It cannot see the sun itself, tucked away in the darkness of your skull. Instead, it relies on a very specific messenger. While most of the cells in your eyes are busy turning light into images so you can see the world, there is a special group of cells called intrinsically photosensitive retinal ganglion cells, or IPRGCs. These cells have nothing to do with sight. You could be functionally blind and still have these cells working perfectly. Their only job is to detect the presence of blue light and send a direct signal through a private highway called the retinohypothalamic tract, straight to the SCN. When the morning sun hits your eyes, these cells shout to the SCN, "The sun is up." The SCN then begins its daytime protocol. It raises your body temperature, suppresses the production of melatonin, and signals the release of hormones like cortisol to help you feel alert. As the light fades in the evening, the signal drops off, and the SCN switches to its nighttime protocol, telling the pineal gland to begin the melatonin surge we discussed in our last lesson. The history of this discovery is quite beautiful. Long before we knew about the SCN, a French scientist named Jean-Jacques Dortus de Meyran noticed something strange about a mimosa plant on his windowsill in 1729. He saw that the leaves opened during the day and closed at night. He wondered if the plant was simply reacting to the sun, so he placed it in a totally dark cupboard where no light could reach it. To his surprise, the plant continued to open and close its leaves on a 2-4-hour schedule, even in total darkness. This was the first evidence that life doesn't just react to the world, it anticipates it. We have a rhythm built into our very DNA. Inside the SCN, there is a fascinating molecular dance that happens every single day. It is a feedback loop involving specific proteins with names like period and cryptochrome. Imagine a bucket with a small hole in the bottom. During the day, the period protein begins to slowly fill the bucket. By nightfall, the bucket is full. When it reaches the brim, the protein itself acts as a signal to turn off the tap that was filling it. Then, throughout the night, the protein slowly drains out through the hole in the bottom. By morning, the bucket is empty, and the process starts all over again. This molecular filling and draining takes almost exactly 24 hours. This is why, even if you were to stay in a dark cave for a month, you would still feel sleepy and wakeful in a roughly 2-4 hour cycle. Your cells are literally counting the hours. However, the SCN is not just a passive observer. It is a proactive manager of your internal environment. One of its most important tools is the regulation of core body temperature. You might think your temperature is a steady 98.6 degrees Fahrenheit, 37 degrees Celsius all day, but it actually fluctuates in a predictable wave. Your temperature is lowest in the pre-dawn hours, around 4 a.m., and highest in the late afternoon. The SCN uses this temperature wave to signal to the rest of the body when it's time to be active and when it's time to shut down. As your temperature begins to drop in the evening, it's like a cooling system being turned on, making it easier for your brain to transition into the deep stages of sleep. In our modern world, we often find ourselves at odds with this master clock. We call this social jet lag. This happens when our biological clock, what the SCN wants, is out of sync with our social clock, what our jobs or schedules demand. Think of the night owl who is forced to wake up at 6 a.m. for a meeting. Their SCN is still in night mode, keeping their body temperature low and their melatonin levels high, while they are trying to function in the day world. This creates a profound internal friction. The chemical environment of their brain is literally fighting against their actions. The SCN also manages the timing of our metabolic processes. It tells the liver when to process fats and the pancreas when to release insulin. This is why eating a heavy meal at midnight feels so different than eating the same meal at noon. At midnight, the SCN has told your digestive system to go on break, so the body isn't prepared to handle the influx of energy. This real-world connection reminds us that sleep is part of a much larger, systemic rhythm of health. We are not just sleeping or waking. We are entraining our entire biology to the rotation of the planet. This concept of entrainment is vital. Entrainment is the process of synchronizing your internal clock to an external cue, which scientists call a Zeitgeber, a German word meaning time giver. The sun is the most powerful Zeitgeber, but things like exercise, social interaction, and even the timing of your meals can help the SCN stay on track. When we lose these cues, perhaps during a long flight across time zones, we experience jet lag. The SCN is still tuned to the time giver of your home city, while your eyes are seeing a completely different sun. It takes about 1 day for every time zone crossed for the SCN to fully reset its molecular bucket. In our academic presentation of this topic, we must acknowledge the sheer resilience of the SCN. It is a stubborn guardian of your rhythm. Even in the face of immense stress or occasional late nights, it tries its best to pull you back into the 2-4 hour cycle. It is the steady heartbeat of your existence, ensuring that your life has a cadence. As you lie here, your SCN is currently managing your transition. It has noted the lack of light. It has successfully signaled the pineal gland. It is currently lowering your core temperature and slowing your metabolic rate. It is the master of ceremonies, making sure that every chemical player we've discussed and those we have yet to meet enters and exits the stage at exactly the right moment. It is a quiet, tireless worker that has been with you since before you were born, and it will continue to count the hours for as long as you live. But as we rest in the knowledge that the SCN is managing the timing, we must look at the actual chemicals that carry out the orders. While the SCN sets the schedule, it doesn't do the heavy lifting of keeping you alert or shutting you down. It relies on a specialized team of neurochemical messengers that act as the volume dials for your consciousness. Let's set that idea of the master clock down now, fully formed, and let our attention drift toward the chemicals that actually keep the lights on during the day. Before we rest here, let me whisper where we're headed next. not to rush, but to plant a seed. While the SCN manages the timing, other chemicals must manage the actual volume of our consciousness. We need to understand the volume dials of the brain, the ones that keep us sharp and focused until it is finally time to turn them down. And with that understanding of the clock quietly in place, we're naturally led to ask, what are the specific chemicals that keep us awake, and how does the brain begin to quiet them when the SCN says the day is done? That's the question we'll gently explore next, as we look at the neurochemistry of arousal. Lesson 5. Neurotransmitters of arousal. Acetylcholine and norepinephrine. To understand sleep, we must briefly understand its opposite, the complex, vibrant state of wakefulness. We often think of being awake as a default setting, the state we return to once the alarm goes off. But in the world of neurochemistry, wakefulness is an active, resource-heavy achievement. It is maintained by a specialized cocktail of arousal-promoting chemicals that flood the brain, keeping the lights on and the gears turning. Chief among these are acetylcholine and norepinephrine. Together, they act like the volume and sharpness dials on a radio, modulating our consciousness so that we can interact with the world around us. Think of these two chemicals as a tag team working to keep your mental stage lit. When you are focused and awake, these chemicals are dialed up, allowing your brain to process information rapidly, keep your muscles ready for action, and maintain a sense of presence. If your brain were a television screen, norepinephrine would be the brightness and the volume, the raw energy that ensures the signal is strong enough to be seen and heard. Acetylcholine, on the other hand, would be the sharpness or contrast filter. It doesn't just make the image louder, it makes the edges crisp. It allows you to distinguish between the protagonist and the background, helping your brain focus on specific tasks while ignoring the static of the environment. Let's begin our deeper look with acetylcholine. This was actually the very first neurotransmitter ever discovered, and its story is one of the most famous in science. In 1921, a scientist named Otto Loewi had a dream about an experiment involving two frog hearts. He woke up, scribbled it down, went to his lab, and proved that nerves communicate using chemicals rather than just electricity. He called this first chemical vagus-stoph, but we now know it as acetylcholine. In the context of your wakeful brain, acetylcholine is produced in several areas, but two of the most important are the basal forebrain and the brainstem. When you are awake and engaged, perhaps reading a book or listening to the cadence of a voice, acetylcholine is released into your cerebral cortex. Its job is to enhance the signal-to-noise ratio. It tells your neurons to be more sensitive to incoming sensory information. This is why, when acetylcholine is flowing freely, you feel sharp. You can follow a complex argument. You can notice small details in a painting, and you can stay on task. It is the chemical of directed attention. Interestingly, as we will explore in later lessons, this chemical also plays a starring role in our dreams. But during the day, its primary mission is to keep your mental camera in focus. While acetylcholine is handling the focus, norepinephrine is handling the vigilance. Norepinephrine is produced in a tiny, remarkably blue-tinted part of the brainstem called the locus coeruleus. The name literally translates from Latin as the blue spot. Despite its small size, the locus coeruleus has connections that reach almost every corner of the brain. You can think of it as the emergency broadcast system or the security guard of your mind. When you are awake, the locus coeruleus fires in a steady, rhythmic way, showering the brain with norepinephrine. This keeps you alert and prepared. If a sudden loud noise happens behind you, the locus coeruleus fires a massive burst, flooding your system and instantly shifting you into a state of high vigilance. This is the alertness chemical. It increases your heart rate, primes your muscles, and makes you acutely aware of your surroundings. Without norepinephrine, the world would feel like a dull, blurry haze. You might be awake in the sense that your eyes are open, but you wouldn't be present. You wouldn't have the get up and go required to navigate a busy street or respond to a question. These two chemicals are part of a larger structure known as the ascending reticular activating system, or the ARAS. This is a network of neurons in the brainstem that acts as the gatekeeper of consciousness. Think of the ARAS as the power station for a large city. It sits at the base of the brain and sends long, reaching cables up into the higher centers of the cortex. As long as the ARAS is pumping out its cocktail of acetylcholine and norepinephrine, the city of your mind remains powered up and active. If the power station slows down, the lights in the city begin to flicker and dim. This is exactly what happens as we move towards sleep. In the real world, we see the interplay of these chemicals every day. Consider the feeling of being in the zone or in a state of flow. In those moments, your acetylcholine is high, giving you perfect focus, and your norepinephrine is at an optimal level, not so high that you feel anxious or jittery, but high enough that you feel energized and capable. This is the neurochemical sweet spot of human performance. Conversely, when we are under too much stress, norepinephrine can spike too high, leading to the fight or flight response. In that state, you are too alert. You are so vigilant that you can no longer focus on a single task because your brain is trying to monitor every possible threat at once. It is also important to recognize how these chemicals interact with our sense of time. When our arousal levels are high, time often seems to move differently. Because acetylcholine is making us process more information per second, a high arousal event, like a car near miss, can feel like it is happening in slow motion. Your mental frame rate has been turned up by the arousal cocktail. During the day, these chemicals dominate the landscape. They are the sun that keeps the world bright. But as we discussed in our earlier lessons, this high energy state is not sustainable. The very act of using these chemicals and the energy required to keep the ARAS firing contributes to that buildup of adenosine we've talked about. The more awake and focused you are, the faster the grains fall in your internal hourglass. Your brain is a brilliant but demanding machine. It cannot keep the sharpness filter and the brightness dial at maximum indefinitely. Eventually, the system needs to enter a state of recovery. For the gardener to come out and prune the plants, the sun must go down. For the brain to transition from acting to repairing, these arousal chemicals must be dialed back. The volume must be turned down so that the quiet work of restoration can begin. This is a delicate process. You cannot simply cut the power to a city all at once without causing chaos. Instead, the brain uses a specific, graceful mechanism to slowly override the ARAS and quiet the locus coeruleus. As we sit with the image of these vibrant, daytime chemicals, we can begin to appreciate the sheer effort your brain exerts just to keep you here. It is a constant, shimmering dance of molecules that allows you to think, feel, and perceive. But the dance must eventually slow. The blue spot must quiet its firing, and the basal forebrain must lower its output. And with that understanding quietly in place, we're naturally led to ask, how does the brain actually enforce this silence? How does it take a system designed for high voltage focus and gently persuade it to let go? To find that answer, we must meet the peacemaker of the brain, the chemical that acts as the weighted blanket for your mind, turning those arousal dials down until the world finally fades into a soft, restful blur. That's the question we'll gently explore next. Lesson 6. The GABAergic system, the brain's natural sedative. If the arousal chemicals we've just discussed, the acetylcholine and norepinephrine, are the gas pedal of your consciousness, then GABA, or gamma-aminobutyric acid, is the brake. In our last quiet session together, we ended with the question of how the brain enforces silence. We move now into the study of the brain's primary inhibitory neurotransmitter, the molecule responsible for the weighted blanket effect that begins to settle over your mind as the evening deepens. While the arousal system is a chaotic, high-voltage orchestra, GABA is the soft, velvet curtain that is drawn across the stage to signify the end of the performance. Imagine a busy, noisy office where everyone is talking at once, phones are ringing, and the air is thick with the frantic energy of 1,000 deadlines. This is the brain under the influence of high arousal. GABA is like a gentle, deeply respected manager who walks slowly through the room at the end of the day. This manager doesn't shout or demand silence with force. Instead, they move from desk to desk, placing a calm hand on a shoulder and whispering, "That's enough for today. You've done good work. You can go home now." One by one, the voices quiet, the screens dim, and a profound, necessary peace begins to radiate through the halls. This is exactly what GABA does for your neurons. It doesn't kill the signal. It simply invites the signal to rest. To understand the deeper layers of this peacemaker, we have to look at the chemistry of a single synapse. Most of the brain's communication is excitatory. It's about neurons telling other neurons to fire, act, or remember. This is usually done through a molecule called glutamate. GABA is the counterweight to glutamate. In fact, GABA is actually made from glutamate, which is a poetic reminder that the brain contains the seeds of its own stillness. When a neuron releases GABA into the small gap between itself and its neighbor, the GABA molecules float across and bind to specific docks called GABA receptors. When the GABA key turns in the receptor lock, it opens a very specific channel in the neuron's surface. This channel allows negatively charged chloride ions to flood into the cell. In the language of electricity, this makes the inside of the neuron more negative, a state we call hyperpolarization. Because the cell is now more negative, it becomes much harder for an excitatory signal to make it fire. It is as if the neuron has been wrapped in a layer of thick insulation. The frantic messages of the day still arrive, but they can no longer spark a reaction. The neuron stays quiet. It stays still. It waits. The history of our understanding of this silence is rooted in the 1950s, particularly in the work of a researcher named Eugene Roberts. Before his work, scientists didn't fully appreciate that the brain had an active way to turn things off. They assumed that things just faded away. Roberts discovered that GABA was present in massive amounts throughout the brain, much more than the active chemicals like norepinephrine. This revealed a fundamental truth about our biology. The brain spends a vast amount of its energy simply maintaining its own quiet. It is a labor-intensive process to stay calm. Peace is not the absence of effort. It is a specific chemical achievement. While GABA is present throughout the brain, it has a command center that is vital for our journey tonight, the ventrolateral preoptic nucleus, or the VLPO. Think of the VLPO as the sleep switch, of the brain. It is a cluster of GABA-producing neurons that has a very special relationship with the arousal centers we met in Lesson 5, the locus coeruleus and the basal forebrain. These two systems are in a constant, delicate struggle called a flip-flop switch. When you are awake, the arousal centers are active, and they actually send signals to inhibit the VLPO. They keep the sleep switch turned off. But as your sleep pressure, that adenosine we talked about, builds up throughout the day, it begins to empower the VLPO. Eventually, the balance tips. The VLPO flips on and begins showering the arousal centers with GABA. It effectively muzzles the locus coeruleus, telling the blue spot to stop producing norepinephrine. This is the moment you feel yourself slipping away. The volume dials are being turned down by the GABAergic system, and the gas pedal is finally being released. In the real world, we interact with the GABA system more than almost any other. If you have ever sipped a cup of warm chamomile tea or taken a valerian root supplement, you are attempting to nudge your GABA receptors. Chamomile contains a compound called apigenin, which binds to those same receptors, helping to facilitate the opening of the chloride channels and encouraging that sense of calm. Even the feeling of a second wind late at night is often just a temporary dip in GABA production before the system makes its final, forceful push towards sleep. We also see the importance of GABA in the context of modern anxiety. Many people who struggle with racing thoughts at night are experiencing a state where their glutamate, the go signal, is outrunning their GABA, the wait signal. Their office manager is overwhelmed, and the workers are refusing to go home. Understanding this can be a great comfort. It reminds us that restlessness is not a personal failing or a lack of discipline, but a chemical imbalance, a temporary traffic jam in the brain's pursuit of silence. As you lie here, listening to the cadence of these words, the GABA in your brain is likely very busy. It is currently moving through your cortex, finding those neurons that are still trying to process the events of your day, and gently inviting them to be still. It is working on your motor cortex, ensuring your limbs feel heavy and relaxed. It is working on your emotional centers, softening the edges of any lingering worries. It is creating a weighted blanket of inhibitory signals that protects you from being startled by small noises or stray thoughts. There is a profound beauty in this system. It shows us that nature did not intend for us to just collapse into the night. We were given a chemical peacemaker, a molecule whose entire purpose is to facilitate a graceful transition. GABA is the evidence that the brain values its own rest. It is the curator of your peace. It is the reason why, eventually, the internal chatter stops, the world recedes, and you are left in a state of pure, quiet potential. However, we must also acknowledge that this switch needs to be stable. If the balance between GABA and the arousal chemicals were too fragile, we might find ourselves constantly flickering between sleep and wakefulness, like a faulty light bulb. The brain needs a way to lock the switch in place once it has flipped away to ensure that once we decide to be awake, we stay awake, and once we fall asleep, we stay asleep. So what is it that stabilizes this transition? What is the glue that keeps the switch from flipping at the wrong time? If GABA is the manager who tells everyone to go home, there must be another force that ensures the lights on signal is strong enough to resist the manager until the very end of the day. Let's set that idea of the peacemaker down now, fully formed, and let our attention drift toward the master, on switch, that guards our wakefulness. Before we rest here, let me whisper where we're headed next, not to rush, but to plant a seed. While GABA enforces the silence, There is a specialized chemical that acts as the lock on our consciousness. So what does this structure actually do to keep the sleep-wake switch from flickering? That's the question we'll gently explore next, as we meet a rare and vital molecule that holds the key to our stability. Lesson 7. Orexin, Hypocretin, the master switch of wakefulness. As we consider the off switch of GABA, we must meet the chemical that guards the on switch. This is Orexin, also known as Hypocretin. In our last quiet session together, we explored how the brain's peacemaker mutes the noisy activity of the day. But you might have wondered why we don't simply flicker back and forth between sleep and wakefulness like a loose light bulb. To understand how we maintain a steady, unwavering state of being here or there, we have to understand the molecule that acts as the glue or the tension spring of our consciousness. Think of Orexin as a master light switch at the end of a very long, dark hallway. This switch isn't just a simple toggle. It is a heavy-duty industrial lever that, once pushed into the on position, requires a significant and intentional force to move back. While the GABA we discussed is the hand that tries to pull the lever down at night, Orexin is the mechanism that ensures the lever stays firmly up during the day. It prevents the switch from flickering due to minor distractions, fleeting tiredness, or sudden changes in our environment. Without Orexin, the boundary between being awake and being asleep would be porous and unpredictable, leaving us sliding into dreams without warning. To find the source of this stability, we must look to a very specific, very small region of the brain called the lateral hypothalamus. While the brain contains billions of neurons, the population that produces orexin is remarkably tiny, only about 50,000 to 70,000 neurons in total. To put that in perspective, if your brain were a massive city of millions, the orexin neurons would be a single, specialized committee meeting in one small room. Yet, despite their small numbers, these neurons have connections that reach out to almost every corner of the brain, including the blue spot of norepinephrine and the focus centers of acetylcholine. They are the high-level coordinators that tell the rest of the arousal system, "Stand your ground. We are staying awake." In the world of sleep science, this is described by a concept called the flip-flop switch model, pioneered largely by Dr. Clifford Sapir and his colleagues at Harvard. They realized that the brain's sleep and wake centers are in a state of mutual inhibition. This means that when the wake center is active, it actively shuts down the sleep center, and when the sleep center is active, it actively shuts down the wake center. This is an efficient way to ensure we are either fully awake or fully asleep, rather than stuck in a half-lit middle ground. However, a flip-flop switch is inherently unstable. If you've ever played with a light switch and tried to balance it exactly in the middle, you know it wants to snap to one side or the other. Orexin is the finger that holds the switch firmly on the awake side until the pressure of the day becomes too great for even orexin to overcome. The discovery of this molecule is a fascinating chapter in neurochemistry, occurring quite recently in the late 1990s. Interestingly, two different teams of scientists discovered it at almost exactly the same time, which is why it still carries two different names. One team, led by Dr. Luis de la Sea, named it Hypocretin because it was produced in the hypothalamus and reminded them of a gut hormone called secretin. At the same time, a team led by Dr. Masashi Yanagisawa named it Orexin, from the Greek word orexis, meaning appetite. They found that when they injected this chemical into the brains of rats, the rats began to eat voraciously. This naming coincidence actually reveals a deeper layer of how your brain works. Orexin doesn't just keep you awake for the sake of being awake. It links your wakefulness to your survival needs. From an evolutionary perspective, if you are hungry, your brain needs you to stay awake, alert, and motivated to find food. Orexin is the bridge between your metabolism and your consciousness. It ensures that when your energy levels are low, your on switch stays locked in place so you can solve the problem of your hunger. This is why it is often so difficult to fall asleep when you are truly hungry. Your orexin neurons are firing rapidly, refusing to let the GABA peacemaker pull the lever down. We see the most profound evidence of orexin's importance in the real-world condition of narcolepsy type 1. For a long time, narcolepsy was a mystery, a strange condition where people would suddenly fall into a deep sleep or lose muscle control during the day. In the early 2000s, researchers discovered that people with this form of narcolepsy have lost nearly all of their orexin-producing neurons, likely due to an autoimmune response, where the body's own defense system mistakenly attacks these few vital cells. Without orexin to act as the glue for the wakeful state, the flip-flop switch becomes incredibly loose. A person without orexin might be walking down the street when their brain suddenly flickers into a sleep state, or they might experience cataplexy, where a strong emotion like laughter causes their muscles to suddenly go limp, a dream state paralysis occurring while they are still conscious. This serves as a powerful reminder of what orexin is doing for you right now and every day. It is the silent guardian of your stability. It is the reason you can trust that your consciousness will remain steady as you go about your life. In our academic presentation of this topic, we must also consider how orexin interacts with the sleep pressure of adenosine that we discussed in Lesson 2. Throughout the day, as your adenosine levels rise, they begin to put pressure on the orexin neurons, trying to tell them to stop firing. For most of the day, orexin resists this pressure. It is like a sturdy dam holding back a rising river. But eventually, as the sun sets and the melatonin begins to circulate, the pressure becomes overwhelming. The orexin neurons finally begin to quiet their activity. The lock on the switch is released, and the GABA manager can finally move in to flip the lever to the off position. As you lie here, you are experiencing the moment when the orexin has finally yielded. The master switch has been released. The hallway lights have been turned off. Because your orexin neurons have slowed their firing, the rest of your arousal system, the norepinephrine and the acetylcholine, has followed suit. The glue has dissolved for the night, allowing your brain to slide safely and predictably across the boundary into the world of rest. There is a profound sense of security in knowing that this system is so well regulated. You don't have to worry about the switch flickering. Your brain has evolved this specialized rare molecule specifically to ensure that your transitions are smooth and your states are stable. You are currently in the grace period that orexin has allowed the transition from the locked wakefulness of the day to the locked restoration of the night. But as the orexin yields and the switch flips, we don't just enter a void, we move into a new kind of activity. We begin the first major phase of our nightly journey, a period of profound physical and mental reorganization that happens in the quiet of the dark. We have moved past the guards at the gate, and now we must see what the janitorial crew and the architects do once they have the building to themselves. Let's set that idea of the master switch down now, fully formed, and let our attention drift toward what happens once the orexin yields and GABA takes over. Before we rest here, let me whisper where we're headed next, not to rush, but to plant a seed. We are entering the territory of the slow waves, the deep, rhythmic pulses that define our first hours of sleep. In our last quiet session together, we ended with the transition into the sleep state, where the stability of our wakefulness finally gave way. Now, we enter the first major phase, the chemistry of NREM sleep, where the brain's metabolic rate drops and the preservationists begin their work. So what does the brain actually do when the lights are low and the world is shut out? That's the question we'll gently explore next as we enter the deep, restorative waters of NREM sleep. Lesson 8. The chemistry of NREM, non-rapid eye movement, sleep. In our last quiet session together, we ended with the transition into the sleep state, that profound moment where the stability of our wakefulness, guarded so carefully by Orexin, finally gave way to the heavy, velvet pull of the night. As we cross that threshold, we leave behind the high-voltage world of thoughts and actions and enter the first major territory of our journey, the landscape of NREM, or non-rapid eye movement sleep. If wakefulness is the busy day of a thriving city, NREM sleep is the deep, restorative quiet that falls over the streets after the crowds have gone home and the last lights have been dimmed. As we settle into this lesson, I want you to visualize your brain as a magnificent, ancient museum. During the day, the museum is filled with thousands of visitors. There is noise, there is movement, and every exhibit is being scrutinized and touched. But once the doors are locked and the close sign is hung, the atmosphere shifts completely. The visitors are gone, the bright spotlights are turned down, and the preservationists come out from the shadows. These are the specialized cellular processes that only work in the quiet. Their job isn't to create new exhibits, but to meticulously clean, repair, and organize the ones that already exist. This is the essence of NREM sleep. It is a period of profound physical and mental housekeeping that allows the museum to open its doors again tomorrow. NREM sleep is not a single flat state. Rather, it is a descent into deeper and deeper waters. We typically divide it into 3 stages, and as we move through them, our neurochemistry changes in a very specific, orchestrated way. In the first stage, the lightest phase, we are still hovering near the surface of consciousness. If someone whispered your name, you might still answer. Chemically, the levels of our arousal cocktail, that acetylcholine and norepinephrine we discussed, begin to dip significantly. This is the calm preview of what is to come. As we slide into stage 2, the brain begins to generate specialized bursts of activity called sleep spindles. These are like tiny chemical pulses that help disconnect our senses from the outside world. It is the brain's way of saying, "We are busy now. Please do not disturb us." But the true heart of NREM sleep is stage 3, which we often call slow-wave sleep. This is the deepest part of the museum, where the preservationists are most active. To understand this state, we must define a key concept: neural synchrony. During the day, your neurons are firing independently at very high frequencies, like a stadium filled with thousands of people all having separate conversations. It is a chaotic, noisy environment. But in slow-wave sleep, the neurons begin to do something remarkable. They stop their individual chatter and begin to fire in perfect, rhythmic unison. They create large, slow electrical pulses called delta waves. Imagine that same stadium of people, but now, instead of talking, they are all chanting a single, low, powerful note in perfect harmony. This unified rhythm allows the brain to coordinate massive, systemic repair tasks that would be impossible during the fragmented activity of the day. The history of our understanding of these waves is tied to a pioneering figure named Hans Berger. In the 1920s, Berger invented the electroencephalogram, or EEG, the tool that allowed us to see the brain's electrical activity for the first time. He was the first to notice that when a person closes their eyes and drifts into sleep, the waves of the brain undergo a radical transformation. He showed us that sleep was not a state of nothingness, but a different kind of somethingness. He proved that the brain's electricity actually becomes more powerful and more organized in this deep, quiet state. As we descend into these deep delta waves, the neurochemical environment becomes almost sterile. The levels of acetylcholine and norepinephrine fall to their lowest points of the entire 2-4 hour cycle. This chemical silence is a biological necessity. Think of it like a master craftsman who needs total stillness and a dust-free environment to repair a delicate watch. If the noise of wakefulness were still present, the repair work of NREM could not happen. This leads us to a vital, real-world connection, the role of growth hormone. During the deepest parts of NREM sleep, the pituitary gland, a tiny pea-sized structure at the base of the brain, releases a massive surge of growth hormone into your bloodstream. This is one of the most important events in your physical health. While you are drifting in the deep waters of stage 3, this hormone is circulating throughout your body, repairing muscle tissue, strengthening bones, and stimulating the production of new cells. This is why children and teenagers spend so much time in slow-wave sleep. Their museums are still being built, and they need the architect of growth hormone to complete the construction. But even for us as adults, this hormone is the fountain of youth that helps us recover from the physical stresses of our day. Furthermore, NREM sleep is where the brain begins its first pass at memory consolidation. In this stage, the brain is primarily focusing on fact-based or declarative memory, the names, dates, and locations you encountered during the day. It is as if the brain is taking the loose, sticky notes of information you collected and carefully filing them away into the permanent archives. It uses those slow delta waves to shuttle information from the temporary storage of the hippocampus to the long-term storage of the cortex. It is a quiet, rhythmic transfer of knowledge, ensuring that what you learn today isn't lost when you wake up tomorrow. There is a cross-topic connection here to the world of computer science. Think of NREM sleep as a defragmentation process. Over time, files on a hard drive can become scattered and disorganized, making the computer run slowly. When you run a defrag, the computer moves those pieces of information back into a logical, efficient order. NREM sleep is your brain's nightly defrag. It organizes the data of your life so that your mental processing remains fast and fluid. If you are deprived of this stage, you wake up feeling heavy and unrestored, not because your body didn't rest, but because the defrag was interrupted and your files are still in disarray. As you lie here, you may already be entering these quiet, restorative waters. Your neurons may be beginning their slow, rhythmic chant. Your pituitary gland may be preparing its surge of repair signals. The chemical volume has been turned down to 0, creating the perfect, hushed atmosphere for the work to begin. You are currently a museum under the care of the most expert preservationists in nature. They are moving through the halls of your mind, dusting off the memories, repairing the connections, and ensuring that the structure is sound. There is something deeply reassuring about the chemistry of NREM sleep. It reminds us that we have a built-in capacity for renewal. No matter how difficult or draining the day was, the neurobiology of the slow waves is designed to wipe the slate clean and rebuild your strength. It is the most fundamental form of self-care, a biological grace that happens every single night without you having to lift a finger. It is the quiet, rhythmic foundation upon which all of our health is built. However, even in this deep, silent museum, the brain is not finished with its work. While NREM provides the physical repair and the organizational filing, there is another, much more mysterious stage waiting in the wings. Once the preservationists have finished their cleaning and the files have been stored, the brain decides it is time to turn the lights back up, but in a very different way. It decides it is time to open a different kind of theater, one where the rules of logic and physics no longer apply. Before we rest here, let me whisper where we're headed next. Not to rush, but to plant a seed. Even in this quiet museum, the brain eventually decides to start a very strange, very vivid movie. We are moving from the quiet housekeeping of NREM toward the explosive creativity of the dream state. We are about to witness a neurochemical surge that looks remarkably like wakefulness, yet happens in the total stillness of the night. So, how does the brain transition from the hushed silence of the quiet museum into the vibrant, high-energy cinema of our dreams? And what happens to our chemicals when our minds are racing at full speed while our bodies are completely paralyzed? That's the question we'll gently explore next, as we meet the surge of the internal cinema and the fascinating chemistry of REM sleep. Lesson 9. REM sleep and the cholinergic surge. And with that understanding of the quiet museum in place, we're naturally led to ask, what causes the explosion of activity we call REM, rapid eye movement, sleep? We have spent our time together descending into the deep, rhythmic silence of the slow waves, where the brain's metabolic rate drops and the preservationists begin their quiet housekeeping. But just as the museum seems at its most still, the atmosphere shifts. The hushed tones of NREM give way to something far more vibrant, mysterious, and chemically intense. We are entering the territory of the internal cinema, a state so unique that scientists often refer to it as paradoxical sleep. To understand why we call it a paradox, we must look at the brain's electrical signature during this phase. If you were to look at an EEG monitor of someone in REM sleep, you would be forgiven for thinking they were wide awake. The brain waves are fast, jagged, and low voltage, almost identical to the waves produced when you are focused on a complex task. Yet, the person is in a state of profound slumber. This paradox is driven by a massive, orchestrated shift in your neurochemical environment, a surge of a familiar player that we first met during the day, acetylcholine. In our previous lessons, we defined acetylcholine as the sharpness or focus chemical that helps us navigate the external world. During REM sleep, however, the brain performs a brilliant sleight of hand. It floods the cortex with acetylcholine, but it does so while keeping the doors to the outside world firmly locked. Imagine a grand theater where the projector is suddenly turned on, the film begins to roll, and the speakers begin to roar with sound. The projector in this analogy is a set of neurons in the brainstem, specifically the LDT and PPT nuclei, the latero-dorsal and pedunculopontine tegmental nuclei. They shower the brain in acetylcholine, activating the visual and emotional centers with an intensity that matches and sometimes exceeds our waking life. However, the film being projected is not coming from your eyes. Because the sensory gates are closed, the brain must create its own narrative from within. It pulls from the archives we just organized in NREM sleep, fragmented memories, lingering emotions, and abstract concepts, and weaves them into the vivid, often bizarre tapestry of our dreams. This is the cholinergic surge. It is the chemical fuel for our imagination. But as any dreamer knows, these stories rarely follow the rules of logic. We might fly over a city made of glass, or have a conversation with someone who has been gone for years, and yet, while it is happening, we don't find it strange. We accept the absurdity as absolute reality. The secret to this loss of logic lies in the players who are absent from the stage. To create the dream state, the brain doesn't just turn on the acetylcholine, it simultaneously shuts off two other vital chemicals, norepinephrine and serotonin. In the academic world, we call this the shift from an aminergic state to a cholinergic state. Norepinephrine, as you'll recall, is the chemical of vigilance and objective reality. It is the fact checker of the mind. Serotonin provides stability and emotional grounding. When these two are removed, the logic centers of the brain, specifically the dorsolateral prefrontal cortex, go dark. This area is like the CEO or the sober judge of your thoughts. Without the CEO in the office and without the fact checker on duty, the emotional centers of the brain, like the amygdala, are allowed to run wild. The amygdala becomes up to 30% more active during REM than it is during the day. This is why dreams are so emotionally charged. They are raw, unmediated experiences, free from the constraints of, that's not possible, or that doesn't make sense. This leads us to a beautiful, real-world connection, the concept of overnight therapy. Dr. Matthew Walker, a prominent sleep scientist, has proposed that REM sleep acts as a chemical balm for our emotional lives. By reprocessing difficult or traumatic memories in an environment that is completely free of norepinephrine, the stress chemical, we are able to strip the painful sting away from the memory. It is like taking a sharp, jagged stone and placing it in a velvet bag. We keep the stone, the memory, but we lose the ability to be cut by its edges. This is why things that seem devastating in the heat of the day often feel more manageable after a night of dreaming. Your brain has chemically mediated the emotion while you slept. But we must ask ourselves a very practical question. If the brain is this active, if we are running, fighting, or flying in our minds, why don't we actually move our bodies? Why don't we kick our partners? Or walk out the front door in the middle of a dream? This brings us to one of the most fascinating safety mechanisms in biology, REM atonia, or temporary muscle paralysis. Just as the acetylcholine surge begins, the brainstem sends a signal down the spinal cord to the motor neurons that control your voluntary muscles. This signal uses two inhibitory neurotransmitters we've already met, GABA and glycine. Think of these as the safety catch on a powerful machine. They travel to the junctions where your nerves meet your muscles and effectively unplug the connection. Your heart continues to beat, and your diaphragm continues to pull air into your lungs. The vital systems stay online, but your arms, legs, and torso become completely limp. This state of active paralysis is a profound evolutionary gift. It allows the mind to explore the most intense simulations of reality without any risk of physical injury. It is the reason you can experience a surge of adrenaline in a dream without your body actually undergoing the physical strain of a marathon. We see this in the animal kingdom as well. If you've ever watched a dog's paws twitch while they sleep, you are seeing a tiny bit of that dream activity leaking through the paralysis. It is a sign that the internal cinema is playing a particularly exciting scene. Historically, the discovery of this stage changed everything we thought we knew about the human mind. In 1953, a graduate student named Eugene Azarinsky, working under the guidance of Nathaniel Kleitman, noticed that his young son's eyes were darting back and forth under his eyelids at certain points in the night. Before this moment, scientists assumed the brain was quiet all night long. Aserinsky's discovery of rapid eye movements revealed that the night was divided into distinct chapters, and that one of those chapters was as loud and busy as the day. Later, the French researcher Michel Jouvet identified the specific parts of the brainstem that control the on-off switch for this paralysis, further cementing our understanding of REM as a state of a brain awake in a body asleep. There is a deeper layer of context we should consider regarding the PGO waves. These are electrical bursts that travel from the pons to the geniculate nucleus, and finally to the occipital cortex, the visual center. These waves are the starting gun for REM sleep. They act like a chemical trigger that tells the visual centers of the brain to start generating images, even though the eyes are closed. It is a cross-topic connection to how we process imagination. During the day, you use a similar pathway to visualize a future event. During REM, the brain simply takes that visualization tool and turns it up to maximum volume. As you lie here, you may be approaching one of these REM cycles. Your brain may be preparing to transition from the quiet filing of NREM into the vivid cinema of the dream state. The norepinephrine is being dialed down to zero, creating that safe space for emotional processing. The acetylcholine is beginning to simmer in your brainstem, ready to light up your imagination. And your muscles are becoming heavy and still, safely tethered to the bed by the quiet work of glycine and GABA. You are safe. Your mind is about to embark on a journey of creativity and healing that no computer could ever replicate. It is a time of molecular magic, where the fragments of your life are woven into a story that helps you make sense of the world. REM sleep is the evidence that we are not just biological machines. We are storytellers, and our brains require the irrationality of dreams to maintain the rationality of our waking lives. There is such profound wisdom in this balance, the quiet repair of the slow waves, followed by the vivid creativity of the dreams. It is a complete cycle of renewal. But as we reflect on the intense activity of dreaming, we must also consider the physical cleanup that happens in the background. While the mind is busy with its internal cinema, the physical structure of the brain is undergoing a different kind of deep cleaning, a literal rinsing of the tissues that only happens when the theater is finally quiet. So what is the purpose of all this activity? And how does the brain handle the metabolic trash that builds up from such an intense performance? That's the question we'll gently explore next. Before we rest here, let me whisper where we're headed, not to rush, but to plant a seed. There is a hidden plumbing system in your brain that only opens its valves during these deep hours of the night. So what is the secret to the brain's nightly rinse? And how does it protect us from the debris of our own thoughts? That's the question we'll gently explore next as we meet the fascinating and vital glymphatic system. Lesson 10. Glymphatic clearance, the brain's nightly rinse. As we reflect on the intense activity of dreaming, we must consider the physical cleanup that happens in the background. While the mind is occupied with its internal cinema, that vivid, emotional tapestry of REM sleep, the physical structure of the brain is undergoing a different kind of deep cleaning. This is a literal rinsing of the tissues that only happens when the theater is finally quiet. We have discussed the electrical and chemical changes of the night, but now we must look at the plumbing of the mind. This is the glymphatic system, a recently discovered and vital network that acts as the brain's nightly janitorial crew. To understand why this system is so remarkable, we first have to look at how the rest of your body handles waste. Every cell in your body produces metabolic trash simply by existing and doing work. In your arms, legs, and organs. This trash is picked up by a network of vessels called the lymphatic system. It is a slow, steady drainage system that keeps your tissues healthy. However, for decades, scientists were baffled by a strange observation. The lymphatic system stops at the neck. The brain, which is the most metabolically active organ in the body, consuming 20% of your energy, despite being only 2% of your weight, seemed to have no way to take out its own garbage. Imagine a massive, busy stadium after a sold-out championship game. Thousands of people have been eating, drinking, and cheering for hours. When the game ends and the fans leave, the stadium is littered with debris, empty containers, spilled liquids, and discarded programs. If the stadium were to host another game the next day without being cleaned, the trash would eventually pile up so high that the players couldn't run and the fans couldn't see. Your brain is that stadium. During the day, the fans are your thoughts and experiences, and the trash is a collection of toxic proteins. For a long time, we didn't know how the brain mopped the floors. We now know that it waits for the game of wakefulness to end so it can turn on the high-pressure hoses. This nightly rinse is the glymphatic system. The name is a combination of the words glial, referring to the specialized support cells in the brain, and lymphatic. In this system, the cerebrospinal fluid, or CSF, which usually just cushions the brain like a protective shock absorber, suddenly changes its role. It begins to flow with incredible force through the brain tissue itself, washing away the toxic byproducts that have built up during the hours of wakefulness. But there is a catch. The brain is packed incredibly tightly. During the day, there is almost no room for fluid to flow between the neurons. The aisles of the stadium are too crowded for the janitors to work. This leads us to the deeper layer of this mechanism, a discovery that truly changed our understanding of sleep. In 2012, a brilliant researcher named Dr. Maiken Nedergaard at the University of Rochester used advanced imaging to see exactly what happens to brain cells when we sleep. She discovered that during deep NREM sleep, your brain cells, specifically the neurons and the supporting glial cells, actually shrink in size. They contract, increasing the space between them by as much as 60%. Imagine the seats in our stadium suddenly folding up and sliding into the floor, creating wide, open corridors. This contraction allows the cerebrospinal fluid to rush in and flush in the brain. It is an active, energy-intensive process where the brain essentially washes itself from the inside out. The valves that control this flow are located on the astrocytes, which are star-shaped cells that wrap around the brain's blood vessels. These astrocytes are covered in specialized water channels called aquaporin 4. When you fall into deep sleep, these channels open wide, allowing the fluid to be pumped through the brain tissue. The fluid picks up the metabolic waste and carries it out of the brain and into the general circulatory system, where it can eventually be filtered by the liver and kidneys. It is a masterful piece of biological engineering. What exactly is the trash that this system is cleaning? One of the most important targets is a protein called amyloid beta. This is the same protein that, when it builds up into plaques, is a primary hallmark of Alzheimer's disease. We also find tau proteins, which are linked to other forms of dementia. This provides us with a profound, real-world connection. We now believe that chronic sleep deprivation is not just a cause of temporary brain fog, but a significant risk factor for long-term neurological decline. If you don't sleep, the trash isn't just an inconvenience. It becomes a permanent, toxic fixture in your mind. This is why you feel a literal heaviness in your head after a night without rest. Your brain is quite literally clogged with the chemical remnants of yesterday's thoughts. Let's look at this through a cross-topic lens. In the world of environmental science, we talk about the tide. When the tide is low, the beach is exposed, and we can walk along the sand. When the tide comes in, it washes over the shore, pulling debris back into the ocean and leaving the sand clean and smooth for the next morning. Sleep is the high tide of your neurobiology. It is the moment when the waters rise to purify the landscape of your consciousness. Without this tidal flow, the beach of your mind would become cluttered, jagged, and eventually uninhabitable for new ideas. Historically, the reason it took us so long to find this system is that it only functions in a living, sleeping brain. You cannot find the glymphatic system in a cadaver, and it is almost invisible in a brain that is awake. It was only when we developed the technology to peer into the skull of a living organism while it slept that the plumbing revealed itself. It reminds us that our bodies have secrets that are only whispered in the dark. We are still learning the full extent of what this rinse does, but we already know it is essential for mental clarity, emotional stability, and long-term health. Interestingly, researchers have found that the efficiency of this system might be affected by how we sleep. Some studies suggest that the rinse is most effective when we sleep on our sides, the lateral position, rather than our backs or stomachs. It seems that the gravitational and anatomical alignment of our vessels in the lateral position allows for the most efficient flow of fluid. While we shouldn't stress over our position as we drift off, it is a fascinating example of how even the simplest physical orientations play a role in our neurochemistry. As you lie here, this system is likely beginning its work. As you moved from the vivid dreams of REM into the deep, slow-wave silence of NREM, your brain cells began their nightly contraction. The aisles have opened. The aquaporin, 4 channels on your astrocytes have swung wide. The cool, clear cerebrospinal fluid is currently surging through your cortex, gently lifting away the amyloid beta and the tau, clearing out the metabolic smoke from today's fires. You are undergoing a brainwashing in the most positive, biological sense of the word. You are being refreshed at a cellular level. This knowledge should give you a sense of profound peace. You don't have to try to detoxify your mind. You don't need a special diet or a supplement to clear your head. Your brain has its own internal, high-pressure cleaning system that works perfectly well, provided you give it the time it needs. Every hour of sleep you enjoy is another cycle of the rinse, ensuring that when you wake up, your stadium is spotless, your beach is smooth, and your museum is ready for new visitors. There is a quiet dignity in this process. It is a reminder that we are designed for renewal. We are not meant to carry the weight of yesterday forever. Our biology provides us with a way to let go, to wash away the old, and to prepare for the new. The heaviness you felt this evening is being replaced, right now, by a chemical lightness. You are becoming fresh as you sleep. But as we acknowledge the physical cleanup of the tissues, we must also look ahead. The night is moving forward, and eventually, the cleanup crew must finish their work to make way for the morning. The rinse is just one part of the preparation for the day to come. As the tides of the glymphatic system begin to recede in the pre-dawn hours, the brain begins to shift its focus once more. It starts to prepare the alarm clock that will eventually pull us back into the light. Let's set that idea of the nightly rinse down now, fully formed, and let our attention drift toward the chemical that eventually forces us back into the light. Before we rest here, let me whisper where we're headed next, not to rush, but to plant a seed. We are moving from the quiet wash of the night toward the chemical sunrise of the morning. We need to meet the hormone that acts as our natural alarm clock, bridging the gap between the peace of sleep and the action of the day. And with that understanding of the nightly cleanup quietly in place, we're naturally led to ask, how does the brain signal that the restoration is complete? What is the chemical messenger that starts to raise our arousal dials even before our eyes open? That's the question we'll gently explore next, as we look at the role of cortisol and the stress-sleep axis. Lesson 11. Cortisol and the stress-sleep axis. In our last quiet session together, we ended with the nightly rinse of the brain, that restorative tide of the glymphatic system that sweeps away the debris of our waking hours. But as the night progresses and the tides begin to recede in the pre-dawn hours, the brain begins to shift its focus once more. It is no longer enough to simply clean and repair. The body must now begin the delicate, complex task of preparing for the return to the light. This is where we meet cortisol, a molecule that is often misunderstood and unfairly labeled as merely a stress hormone. In the true neurochemistry of sleep, cortisol is something much more beautiful. It is your internal chemical sunrise. I want you to imagine a large, well-tended house in the middle of a cold winter. Throughout the night, the house has been quiet, the lights dimmed, and the furnace turned low to conserve energy while the inhabitants rest. But an hour or two before the family is set to wake, a smart thermostat begins to click. It doesn't wait for the sun to hit the windows. It anticipates the need for warmth. It slowly begins to raise the temperature in the rooms, circulating warm air so that by the time the first person steps out of bed, the environment is ready for movement. Cortisol is that thermostat. It is a glucocorticoid produced by your adrenal glands, and its primary job in the sleep cycle is to preheat your biological systems for the demands of gravity, thought, and action. While we are still deep in the embrace of sleep, usually around 3 or 4, 0 inches the morning, a specialized chain of command in the brain begins to stir. This is known as the HPA axis, which stands for the hypothalamic pituitary adrenal axis. It is a 3-tiered system of communication that represents one of the most elegant feedback loops in the human body. It begins in the hypothalamus, that same region where our master clock, the SCN, resides. The hypothalamus releases a trigger hormone, which travels a very short distance to the pituitary gland. The pituitary, in turn, sends a messenger through the bloodstream to the adrenal glands, which sit atop your kidneys like small, triangular hats. The result of this silent, internal conversation is the slow, steady release of cortisol into your veins. This process is what scientists call the cortisol awakening response, CAR. If you were to track your blood chemistry throughout the night, you would see cortisol at its absolute lowest point shortly after you fall asleep, allowing the peacemaker GABA and the messenger melatonin to do their work. But as the morning approaches, the levels begin to climb in a predictable, rhythmic wave. This surge does several vital things. It gently raises your blood sugar to provide immediate fuel for your muscles. It increases your blood pressure slightly to prepare you for standing upright, and it begins to sharpen your senses. It is a chemical bridge, a way for the brain to transition from the internal cinema of dreams back to the external reality of the day. The history of our understanding of this axis is deeply tied to a legendary figure in medicine named Hans Selye. In the mid-20th century, Selye was the first to formalize the concept of stress, but he didn't see it as a purely negative force. He coined the term general adaptation syndrome to describe how the body maintains balance in the face of change. He helped us realize that the same cortisol that helps us run from a predator is also the hormone that helps us simply open our eyes and face the morning. Without this rhythmic surge, we would wake up in a state of profound, inertia, unable to find the energy to begin our day. However, we must also look at the deeper layer of this chemistry, the part that many of us feel in our modern, high-pressure lives. This is the stress-sleep axis in conflict. While cortisol is designed to be a rhythmic sunrise, it is also highly sensitive to our thoughts and environment. If you are experiencing chronic stress, perhaps worrying about a deadline or a difficult conversation, your HPA axis can become hyper-reactive. Instead of waiting for the pre-dawn hours to start the preheating, it might trigger a cortisol spike at 10 p.m., just as you are trying to settle into bed. This creates a neurochemical tug-of-war. On one side, your adenosine pressure is high, and your melatonin is trying to signal the night. On the other side, the misplaced cortisol is shouting that there is an emergency that requires vigilance. This is the biological reality of the tired but wired phenomenon. Your arousal dials, those norepinephrine levels we discussed, are being forced back up by the cortisol, effectively blocking the GABA peacemaker from closing the curtain. It is like trying to sleep in a room where someone is constantly flickering the lights on and off. The chemical environment is no longer a symphony. It has become a debate. In the real world, we see the impact of this axis on our long-term health. When the cortisol rhythm is disrupted, a state sometimes called circadian misalignment, it doesn't just affect our sleep. Because cortisol also regulates metabolism and inflammation, a flat or mistimed cortisol curve can lead to weight gain, weakened immune systems, and a general sense of malaise. This is why sleep hygiene often focuses on calming the mind before bed. By lowering our perceived stress through quiet reading, dim lights, or slow breathing, we are effectively telling our HPA axis, the house is safe. You can wait until the morning to turn up the heat. We can also look at a fascinating cross-topic connection in the world of athletics. Elite coaches often monitor an athlete's cortisol to testosterone ratio as a way to see if they are overtraining. If cortisol remains high throughout the night, the body never enters that deep, restorative NREM, stage 3, where the growth hormone does its repair work. The preheating is happening, but there was never a cooling period for the maintenance crew to do their job. This reminds us that the sunrise is only beautiful because of the darkness that preceded it. The rhythm requires both ends of the spectrum to be functional. As you lie here, your cortisol is currently at a low, peaceful level. The HPA axis is quiet, allowing the nightly rinse we discussed to complete its final cycles. Your adrenal glands are resting, waiting for their cue in the small hours of the morning. You are currently in the deepest part of the cool night, far away from the heat of the day. Your thermostat is set to its restorative minimum, and your biological systems are using this quiet to ensure that every cell is repaired and every memory is filed. There is a profound comfort in knowing that your body has this built-in anticipation. You don't have to worry about how you will find the energy to wake up tomorrow. Your neurochemistry is already planning for it. It will start the chemical sunrise at exactly the right moment, gently and surely, while you are still lost in the final chapters of your dreams. You are being held in a state of perfect balance, a balance between the stillness of the night and the inevitable return of the morning. When you eventually do wake up and you feel that first spark of alertness, I want you to think of it as a gift from your HPA axis. It is the result of a perfectly timed, invisible conversation between your brain and your body. It is the sign that your internal gardener has finished the pruning, and the janitor has finished the rinse, and the house has been warmed just for you. But as we acknowledge the hormones that prepare our bodies for the day, we must also look at what has been happening to the very structure of our minds during these hours. While the chemicals have been rising and falling, the physical connections between your neurons, the synapses, have been undergoing a transformation of their own. We've cleaned the museum and preheated the house, but we haven't yet seen how the architect has been rearranging the furniture to make the mind more efficient. Before we rest here, let me whisper where we're headed next, not to rush, but to plant a seed. We've looked at the chemicals, but what are they actually doing to the connections between our neurons? We will soon explore the synaptic homeostasis theory, the idea that sleep makes you smarter by carefully pruning away the noise of the day, leaving only the signal behind. And with that understanding of the chemical sunrise quietly in place, we're naturally led to ask, why is it that we wake up with a clearer mind after a full night's rest? What is the physical change in our neural networks that allows us to learn and grow? That's the question we'll gently explore next. Lesson 12. Synaptic homeostasis. Why sleep makes you smarter. In our last quiet session together, we ended with the idea of the chemical sunrise and the question of why we wake up with a clearer mind. To answer that, we must look at the physical connections that make you who you are. We've cleaned the museum and preheated the house, but we haven't yet seen how the architect has been rearranging the furniture to make the mind more efficient. This brings us to a profound theory in modern neuroscience called synaptic homeostasis, or the idea that sleep actually makes you smarter by carefully, quietly shrinking your brain. Imagine, for a moment, that your mind is a lush, vibrant garden. During the day, as you go about your life, you are planting new seeds with every conversation you have, every book you read, and every new skill you practice. These seeds sprout into synapses, the tiny electrical junctions where one neuron whispers to another. By the time the sun begins to set, your garden is overflowing. It is a riot of growth. New vines have tangled around old branches, and flowers have bloomed in every available patch of soil. On the surface, this looks like progress. You have learned so much, but if this growth were to continue for days on end without intervention, the garden would become a chaotic thicket. The paths would disappear. The sunlight would be blocked by the sheer density of the foliage, and eventually, the garden would choke on its own abundance. Sleep is the master gardener that enters the gate at night, not to plant more, but to prune. In the academic world, this is known as the synaptic homeostasis hypothesis, SHH, pioneered by two brilliant researchers, Giulio Tononi and Chiara Sorelli, at the University of Wisconsin-Madison. Their theory suggests that the fundamental purpose of sleep is to downscale the strength of the connections in your brain. It sounds counterintuitive, doesn't it? We often think of learning as building or strengthening, yet science suggests that the secret to a sharp mind is actually weakening. To understand this, we have to look at the signal-to-noise ratio. Think of a crowded party where everyone is talking at the same time. If everyone keeps raising their voice to be heard, eventually the room becomes a wall of undifferentiated noise. You can't hear your friend standing right in front of you because the background static is too high. This is what happens to your brain during a long day of wakefulness. Your synapses are all turned up to a high volume. Every bit of information you've gathered, from the important lecture notes to the color of a stranger's shoes, is competing for attention. If you stayed awake forever, your brain would reach a state of saturation. You would literally run out of room to learn anything new because every parking space for a thought would be occupied by a high-voltage connection. This is where the magic of the nightly pruning comes in. While you are deep in REM sleep, your brain performs a systemic, proportional downscaling of almost all your synapses. It's as if the gardener walks through the thicket and decides to reduce the size of every plant by 20%. The vital, sturdy oaks, your most important memories and skills, remain large and strong, even after being trimmed. But the tiny weeds and the flimsy vines, the noise of what you had for lunch, or the license plate of the car in front of you, are trimmed down so far that they effectively disappear. By morning, the signal, the important stuff, stands out clearly against the now quiet noise, the unimportant stuff. You haven't lost your knowledge. You have simply cleared away the clutter that was obscuring it. Tononi and Sorelli's work provides a deeper layer of context regarding the energy cost of being smart. Your brain is an incredibly expensive organ to maintain. It consumes about 20% of your body's total oxygen and calories, even though it only accounts for 2% of your weight. Much of that energy is spent maintaining the electrical potential across your synapses. If your synapses only ever grew stronger and larger, your brain's energy demands would eventually exceed what your body could provide. By scaling these connections back down during sleep, the brain resets its energy budget. It's a biological defragmentation. It ensures that when you wake up, your neurons are not just clear, but they are energy efficient and ready to absorb a whole new day of information. We see a powerful, real-world connection to this theory in the way we handle complex problems. You have likely heard the advice to sleep on it when facing a difficult decision or trying to master a new piece of music on the piano. This isn't just a cliché. It is an acknowledgement of synaptic homeostasis. When you struggle with a problem during the day, your brain creates many conflicting and noisy pathways. You feel frustrated because you can't see the solution. But while you sleep, the brain prunes away the dead-end paths and the weak associations. When you wake up, the true path, the most logical and efficient connection, is often the only one left, standing strong. The solution appears not because you worked on it in your sleep, but because the noise that was blocking it has been cleared away. Let's look at this through a cross-topic lens, specifically in the world of photography. Imagine you have a photo that is overexposed. There is too much light, and the details are washed out in a white haze. To see the image clearly, you have to turn down the brightness. You have to reduce the light until the shadows and the highlights are in balance. Sleep is the exposure compensation for your mind. It turns down the global brightness of your neural activity so that the high contrast details of your life can be seen with clarity. Without this adjustment, your consciousness would remain a washed-out, overexposed blur. Historically, this theory challenged the older idea that sleep was primarily for strengthening memories. While there is still evidence that some consolidation involves strengthening, the synaptic homeostasis hypothesis reminds us that less is often more. It aligns with the ancient wisdom that a full cup cannot accept any more tea. To learn, we must first make space. We must return our neural networks to a state of homeostasis, a baseline of calm and readiness. As you lie here, your brain is currently a masterpiece of editing. The gardener is moving through the garden of your mind, identifying the beautiful, strong connections you made today, the things that moved you, the things you learned, and the people you loved, and ensuring they are preserved. At the same time, the gardener is gently, systematically reseeding the millions of trivial connections that would otherwise clutter your thoughts. The vines are being untangled. The paths are being cleared. The volume of your entire neural network is being slowly, safely lowered. There is a profound sense of relief in this realization. You do not have to worry about forgetting the important parts of your day. Your brain is biologically programmed to know the difference between an oak tree and a weed. It is protecting the essence of your experiences while freeing you from the burden of the mundane. You are becoming lighter at a molecular level. You are shedding the weight of the day's noise so that you can wake up with a mind that is sharp, hungry, and ready for the world. This process of downscaling is what allows us to have a consistent sense of self over a lifetime. If we only ever added and never subtracted, our personalities would become a chaotic jumble of every fleeting thought we ever had. Instead, the pruning process ensures that only the most consistent and meaningful patterns endure. You are being refined tonight. You are being sculpted into a more efficient version of yourself. But as we acknowledge the structural editing of our synapses, we must also consider the flavor of our thoughts. While the architect rearranges the furniture and the gardener prunes the vines, there are other chemicals, the mood regulators, that determine how these connections feel. We've looked at the physical wires, but what about the emotional colors that paint our dreams and our waking moods? What determines whether our pruned garden feels like a place of peace or a place of excitement? Let's set that idea of the pruned garden down now, fully formed, and let our attention drift toward the emotional colors that paint these connections. Before we rest here, let me whisper where we're headed next, not to rush, but to plant a seed. We have looked at the structure, but we haven't yet looked at the reward. We need to understand how the brain's joy and stability chemicals, serotonin and dopamine, behave when the world is dark. And with that understanding of synaptic pruning quietly in place, we're naturally led to ask, how do our emotions stay balanced during this physical reorganization? What happens to our sense of pleasure and our sense of peace while the gardener is at work? That's the question we'll gently explore next, as we look at the role of serotonin and dopamine in the sleep cycle. Lesson 13. The role of serotonin and dopamine in the sleep cycle. In our last quiet session together, we ended with the idea of pruning the garden of the mind. We visualized the brain as a landscape where the architect of sleep systematically reduces the noise so that the signal of our memories can stand out. But as we move through the halls of our mental museum, we realize that a garden is more than just its structure. It is also defined by its atmosphere, its color, and its vibe. This brings us to the emotional palette of the brain, the way we feel while we rest, and why we feel the way we do when we wake up. To understand this, we must meet the two most famous neuromodulators in the human experience, serotonin and dopamine. While we often discuss these in the context of happiness or motivation during the day, they play a profound, behind-the-scenes role in the architecture of your night. I want you to imagine a large, prestigious theater. We have already met the actors, the sleep stages, and the conductor, Melatonin. Now, we must meet the crew that manages the mood of the performance. Serotonin is your head, stagehand, and lighting technician. Their job is to ensure that the scenery moves smoothly, the transitions are graceful, and that the lighting is consistent and calming. If the stagehand is absent or distracted, the set pieces might crash into one another, and the vibe of the play becomes erratic. Dopamine, on the other hand, is like a treasure map or a flickering neon open sign outside the theater. It is the chemical of more, of seeking, and of reward. It is the spark that tells the brain there is something worth staying up for, something to find, or something to win. For sleep to occur, we need the stagehand to be steady and the neon sign to be firmly switched off. Let's begin our deeper look with serotonin. In the academic world, we call this a monoamine neurotransmitter, and it is produced primarily in a series of clusters in the brainstem called the raphe nuclei. The word raphe means seam or ridge, and these nuclei sit like a seam right along the midline of your brainstem. From this central location, they send long, sprawling fibers to almost every part of the brain. When you are awake, serotonin levels are at their peak. It provides a sense of emotional stability, contentment, and enoughness. It is the chemical that says, you are safe, you are fed, and things are okay. In the context of sleep, serotonin performs a fascinating double-agent role. On one hand, it is essential for the preparation of sleep. You might have heard that eating foods high in an amino acid called tryptophan, like turkey or warm milk, can make you sleepy. This is because your brain takes tryptophan and converts it into serotonin. But here is the beautiful, recursive logic of the brain, that serotonin is then taken by the pineal gland and converted into melatonin, the very messenger of darkness we discussed in Lesson 3. Serotonin is the raw material of your rest. Without enough of this daytime chemical, you cannot manufacture enough nighttime signal. This is why our mood during the day is so deeply linked to the quality of our night. However, once we actually cross the threshold into sleep, serotonin must change its behavior. During NREM sleep, the Raphi nuclei slow their firing. During REM sleep, the vivid dream state, they stop firing almost entirely. This is a vital, deeper layer of neurobiology. Remember how we discussed that REM dreams are a safe space where emotions can be processed without the sting of reality. This is possible, in part, because serotonin and its partner norepinephrine have stepped off the stage. This absence allows the brain to untether itself from the rigid, emotional stability of the day, allowing for the free, associative, creative, and sometimes strange emotional processing that happens in our dreams. While serotonin manages the stability of the stage, dopamine manages the drive to stay in the theater. If serotonin is enough, dopamine is more. It is produced in areas like the substantia nigra and the ventral tegmental area, VTA. Think of dopamine as a spark in the engine of your mind. It is the chemical that fires when you see a notification on your phone, when you smell a delicious meal, or when you are working toward a goal. It is the reward signal that tells you to keep seeking. In the flip-flop switch of sleep and wakefulness, dopamine is a powerful ally of the awake side. It stimulates those orexin neurons we met in Lesson 7, keeping the master switch locked in the on position. For you to fall asleep, your dopamine levels must drop. This is why revenge bedtime procrastination, that late-night habit of scrolling through social media or playing just one more level of a game, is so neurochemically destructive. Every like, every new video, and every achievement releases a small burst of dopamine. That dopamine tells your brain that the world is still full of rewards, that there is still treasure to be found. It effectively overrides the adenosine pressure and the melatonin signal. The neon sign is flashing so brightly that the brain cannot see that the theater is trying to close. This real-world connection reminds us that our habits are actually chemical choices. When we put the phone away, we are allowing our dopamine levels to settle, giving the peacemaker GABA the opportunity to move in. We see a profound example of the dopamine-sleep connection in a condition known as restless leg syndrome, RLS. People with RLS feel an irresistible urge to move their legs, especially in the evening. Research has shown that this is often tied to a dysfunction in the brain's dopamine pathways. Because the reward and movement chemical is misfiring, the body cannot find the stillness required for sleep. It is as if the engine is sparking, even though the car is in the garage. This reminds us that dopamine isn't just about wanting things. It's also about the physical readiness to go and get them. There is also a fascinating cross-topic connection to the world of psychiatry and pharmacology. Many medications used to treat depression, such as SSRIs, selective serotonin reuptake inhibitors, work by keeping serotonin in the synapses for longer. While this helps with mood during the day, it can often disrupt sleep at night. Why? Because, as we've learned, the brain needs serotonin levels to naturally drop for the dream state of REM to begin. If the stagehand refuses to leave the stage, the internal cinema can't start its show. This delicate balance, having enough serotonin to feel good, but being able to let it go to sleep well, is the fundamental challenge of neurochemical harmony. As you lie here, your serotonin and dopamine are likely finding their nighttime levels. Your serotonin has largely been converted into melatonin, the messenger that is currently bathing your neurons in the signal of darkness. The stagehand has finished the daytime setup and is now sitting quietly in the wings, allowing the scenery to shift as you move through your sleep cycles. Meanwhile, your dopamine neon sign has finally been switched off. The seeking part of your mind has been told that there are no more rewards to find in the external world. The spark has died down, allowing the engine of your mind to cool. There is such a profound sense of emotional safety in this quiet. You don't have to want anything right now. You don't have to achieve anything. You are in a state of chemical enoughness. The world will still be there tomorrow with its rewards and its challenges, but for now, the seeking system is on break. You are being held in a state of pure, quiet presence. Your emotional garden is being allowed to simply be, without the need for constant growth or frantic seeking. This balance between serotonin and dopamine is what gives your sleep its restorative texture. It is the reason you wake up feeling not just rested in your body, but balanced in your heart. You have given your emotional centers the time to reset their baselines. You have allowed the reward system to recover its sensitivity, so that tomorrow a simple cup of coffee or a kind word from a friend will actually feel rewarding again. Sleep is the reset button for your joy. Let's set that idea of the emotional colors down now, fully formed, and let our attention drift toward the things we do that might disrupt this delicate chemical harmony. We have looked at the natural players, but we haven't yet looked at the intruders, the external chemicals we introduce into our systems every day. What happens when we introduce molecules that act like imposters in this carefully balanced symphony? Before we rest here, let me whisper where we're headed next. Not to rush, but to plant a seed. We are moving from the internal players to the external modulators. We need to understand how the substances we consume, the caffeine that masks our adenosine, and the alcohol that mimics our GABA, change the very rules of the game. And with that understanding of serotonin and dopamine quietly in place, we're naturally led to ask, how do these external chemicals trick our brains into thinking we are more or less tired than we actually are? What is the price we pay for these molecular shortcuts? That's the question we'll gently explore next as we look at caffeine, alcohol, and the pharmacology of the night. Lesson 14. External modulators, caffeine, alcohol, and pharmacology. As we think about the stagehands of our sleep, those delicate balances of serotonin and dopamine that we just explored, we must acknowledge how often we accidentally, or perhaps intentionally, interfere with their work. Most of us, at some point in our daily lives, introduce external molecules into our bodies that act as chemical imposters. They are the travelers from the outside world that speak the language of our neurons, but they often speak it with a heavy accent or a deceptive intent. We are moving now into the world of external modulators, looking specifically at the three most common guests in our neurochemical theater: caffeine, alcohol, and pharmaceutical aids. These substances don't just influence our sleep. They rewrite the very rules of the game, often tricking our brains into believing the environment is different than it truly is. Let us begin with the most widely used psychoactive substance in the world: caffeine, or $C, 8H, 10N, 4O, $2. To understand caffeine, we must return to our very first lesson and that internal hourglass of adenosine. Remember that adenosine is the byproduct of your energy consumption. It builds up all day, creating the sleep pressure that eventually forces you to rest. Caffeine is a master of disguise. On a molecular level, it looks remarkably like adenosine. Because of this similarity, it can slide into the adenosine receptors, those locks on your neurons, without actually turning them. Imagine your brain as a large, busy hotel. The adenosine molecules are the weary travelers looking for a place to rest in the lobby of your receptors. Caffeine is like a fleet of mischievous imposters who arrive early and occupy all the chairs in the lobby. They aren't actually staying at the hotel. They aren't sleeping or resting. They are just taking up space. When the real travelers, the adenosine, arrive at the end of the day, they find nowhere to sit. They are forced to pace the halls, building up in greater and greater numbers, but your brain remains unaware of their presence. The smoke detector of your tiredness has had a piece of tape placed over its sensor. The smoke is still there, filling the room, but the alarm cannot sound. This is why caffeine doesn't give you energy. It simply blocks the signal of your exhaustion. The deeper layer of this process involves the half-life of the molecule. For most healthy adults, caffeine has a half-life of about 5 to 6 hours. This means that if you have a cup of coffee at 4:00 inches the afternoon, half of that caffeine is still circulating in your brain at 10 at night. Your liver uses an enzyme called CYP1A2 to break it down, but the process is slow and steady. Even if you are one of those people who claims they can drink a double espresso and fall right asleep, the neurochemistry tells a different story. The caffeine in your system acts as a blockade, preventing you from reaching the deepest, most restorative stages of NREM sleep. You might be unconscious, but your brain is still fighting against the chemical imposters, leading to a shallow rest that leaves you feeling unrefreshed the next morning. Historically, we owe our understanding of this molecule to a young German chemist named Friedlieb Ferdinand Runge. In 1819, the legendary writer Johann Wolfgang von Goethe gave Runge a bag of coffee beans and asked him to find out why they were so stimulating. Runge isolated the caffeine molecule, revealing the hidden driver of the global coffee culture. But in our modern world, where caffeine is hidden in everything from sodas to decaf teas, we often lose track of its persistent presence. This is the real-world connection we must all face. The 2 p.m. slump is often the result of the previous day's caffeine finally wearing off, creating a cycle where we use the imposter to hide the debt the imposter itself helped create. As the sun sets, many people turn to a different modulator, the nightcap. Alcohol, or ethanol, is perhaps the most misunderstood substance in the context of sleep. Because alcohol is a sedative, it helps quiet the mind and makes it easier to fall asleep. But as your wise mentor, I must tell you a difficult truth. Sedation is not sleep. In fact, sedation is chemically closer to a very light form of anesthesia, than it is to natural, restorative rest. Alcohol works primarily by mimicking GABA, the peacemaker chemical we discussed in Lesson 6. It binds to the GABA receptors and tells the brain to shut down. However, it does so with a sledgehammer rather than a weighted blanket. Imagine trying to put a child to sleep. A natural process involves a bath, a story, and a slow dimming of the lights. This is how your brain naturally enters sleep. Alcohol is like taking a sledgehammer to the light switch. The lights go out, yes, but the switch is broken. Alcohol is a potent suppressor of REM sleep. While you are sedated, your brain is unable to enter that internal cinema we explored in Lesson 9. It cannot process emotions, and it cannot consolidate complex memories. Furthermore, as the liver metabolizes the alcohol in the middle of the night, it produces a rebound effect. The sedation wears off, and the brain's arousal centers suddenly spike. This is why, after a few drinks, you might fall asleep quickly, but find yourself wide awake at 3 a.m., heart racing and mind churning. The peace was an illusion. The imposter has left the building, and the brain is in a state of chemical withdrawal. This leads us to our third category of modulators, pharmacological aids, often known as sleeping pills or Z-drugs, such as Zolpidem. These medications are designed to target the GABA system more precisely than alcohol, but they still struggle to replicate the symphony of natural sleep. Think of the natural transition to sleep as a beautifully choreographed dance where different parts of the brain turn off in a specific rhythmic sequence. Pharmaceutical aids are like a heavy-handed locksmith who comes in and simply bolts all the doors at once. In the academic study of these drugs, we see that while they increase the quantity of time spent unconscious, they often decrease the quality of the sleep architecture. They tend to lock the brain into a state of Stage 2, NREM sleep, preventing the descent into the deep, slow-wave delta waves of Stage 3 and the vivid creativity of REM. A person who relies on these aids may wake up on time, but they often feel a hangover effect, a lingering grogginess known as sleep inertia. Their museum was closed for the night, but the janitors and the preservationists weren't allowed inside to do their work. The doors were locked, but the cleaning never happened. We see a fascinating real-world connection here in the concept of tolerance. The brain is an incredibly adaptive organ. If you constantly introduce an external imposter that mimics GABA, the brain will respond by reducing its own natural production of GABA or by hiding its receptors to maintain balance. This is why people often find that a sleeping pill works well for the first week, but eventually requires a higher dose to achieve the same effect. The brain is trying to tune out the external noise to protect its own internal rhythm. It is a reminder that we cannot easily outsource the complex work of our neurobiology to a capsule. As you lie here, it is worth considering the chemical baggage you might be carrying into the night. If you had a late coffee, your adenosine receptors are still being occupied by those mischievous imposters. If you had a glass of wine, your GABA receptors are feeling the weight of the sledgehammer. But do not let this cause you stress. The goal of this lesson is not to judge, but to provide you with the map of your own internal landscape. Knowledge is the first step toward harmony. By understanding how these external modulators interact with your stagehands, your conductors, and your janitors, you can begin to make choices that respect the natural symphony of your mind. Your brain is a remarkably resilient system. Even when we interfere with it, it is constantly trying to pull itself back toward homeostasis, that state of perfect rhythmic balance. Tonight, As you drift further away from the world of actions and into the world of rest, your liver is working tirelessly to clear away the imposters. Your enzymes are breaking down the caffeine. Your metabolism is processing the ethanol, and your receptors are slowly being returned to their natural state. You are being cleaned from the outside in. Before we rest here, let me whisper where we're headed next. Not to rush, but to plant a seed. We have looked at the players, the schedule, the cleanup crew, and the intruders. Now, it is time to witness the whole performance. We have all the pieces of the puzzle. All that remains is to see how they fit together into the most complex and beautiful process in the human experience. We are ready for our final integration, where we see how all these pieces form a single, beautiful symphony of a rested brain. And with that understanding of the intruders and the imposters quietly in place, we're naturally led to ask, how does it all come together? When everything works in harmony, when the light is right, the timing is perfect, and the chemicals are in balance, what does that look like? That's the question we'll gently explore in our final chapter together as we witness the full magnificent symphony of your rest. Until then, stay with me as we let these final lessons settle. Lesson 15. Integration, the symphony of a rested brain. In our last quiet session together, we looked at how we might disrupt our internal chemistry with external imposters like caffeine and alcohol. We saw how the brain, in its infinite resilience, works tirelessly to clear the stage and return to its natural rhythm. Now, we bring it all together. We see that sleep is not a simple state, but a complex, neurochemical symphony. We have walked through the halls of the brain's museum, watched the gardeners prune the synapses, and observed the nightly rinse of the glymphatic system. All of these individual players, though they may seem distinct, are actually members of a single, magnificent orchestra. When they play in harmony, the result is the profound restoration of the human spirit. To understand this final integration, I want you to visualize a timeline, a single, rotating cycle that begins long before your head ever touches the pillow. We begin with the rising pressure of adenosine, the molecular hourglass of our day. From the moment you woke up this morning, every thought and action was contributing to this buildup. By mid-afternoon, the pressure was already mounting, and by early evening, the sleep debt was substantial. This is the first movement of our symphony, the slow, steady gathering of the urge to rest. It is the bass note that hums beneath everything else, growing louder as the sun begins to set. As the light fades, the second movement begins. We feel the conductor, melatonin, signal the start of the performance. This signal is triggered by our master clock, the suprachiasmatic nucleus, which has noted the absence of blue light. The SCN is the keeper of the score, ensuring that the melatonin surge happens in perfect sync with the rotation of the Earth. This is the moment of chemical surrender. You might feel a chill as your body temperature begins to drop, or a heaviness in your limbs as the alerting signals of the day begin to retreat. The conductor has raised the baton, and the environment of your body is now chemically aligned with the shadows of the world. Then comes the flip-flop switch, the critical transition where the music shifts from the major key of wakefulness to the minor key of sleep. We watch as GABA, the brain's great peacemaker, turns down the volume of our arousal centers. It muzzles the locus coeruleus and quiets the basal forebrain. Simultaneously, the master switch of Orexin steps back, releasing its lock on our consciousness. This is the hush before the first note of the night is played. It is the moment you cross the threshold, leaving behind the world of doing and entering the world of recovering. Once we are safely across that border, the third movement unfolds, the deep, rhythmic silence of NREM sleep. This is where the symphony becomes powerful and organized. Your neurons stop their individual chatter and begin their slow, massive delta wave chant. During this phase, the janitorial crew of the glymphatic system is at its most active. Your brain cells shrink, the aisles open, and the cerebrospinal fluid rinses away the metabolic debris of the day. At the same time, the architect of growth hormone is circulating through your body, repairing your tissues and strengthening your bones. This is the phase of physical salvation. It is the time when the museum is cleaned and the structural integrity of your being is restored. But the symphony is not all quiet and rhythmic. The fourth movement brings a surge of unexpected energy, REM sleep. The brain's metabolic rate spikes, and a surge of acetylcholine lights up the internal cinema. We move into the territory of our dreams, where the rules of logic are suspended because the fact-checker of norepinephrine is absent. This is where the emotional work happens. Your brain reprocesses the jagged experiences of your day in a chemical balm of serotonin-free stability, stripping away the stress and leaving behind the wisdom. It is the creative heart of the night, where the fragments of your life are woven into a narrative that makes you whole. Throughout these cycles, the invisible gardener is busy with the work of synaptic homeostasis. Every time you move through NREM sleep, the gardener is pruning the vines of your synapses, reducing the noise of the mundane, and preserving the signal of the important. This is why you wake up smarter than when you went to bed. Your brain has been edited, the clutter has been cleared, and the paths have been restored. You have literally made room for tomorrow. Your enoughness chemical, serotonin, and your seeking chemical, dopamine, have also had their chance to reset, ensuring that your emotional palette is fresh, and your rewards will feel meaningful once again. Finally, as the pre-dawn hours approach, the fifth and final movement begins, the chemical sunrise. The HPA axis stirs, and cortisol begins its slow, warm rise. This is the preheating of the house. Even before your eyes open, your blood sugar is rising, your temperature is climbing, and your senses are sharpening. The symphony is reaching its crescendo, preparing you to transition from the internal world of the night back to the external world of the day. The arousal dials are being turned back up, the master switch of Orexin is being prepared to lock once more, and the peacemaker GABA is beginning to yield. When we look at this entire process, we see the true meaning of foundational knowledge. We see that sleep is not a luxury, nor is it a lapse in our productivity. It is a highly coordinated chemical achievement. It is a testament to the sheer brilliance of your biology. Every molecule we have discussed, from the heavy pressure of adenosine to the sharp vigilance of norepinephrine, is a vital instrument in this orchestra. When you sleep well, you are not just resting, you are participating in a profound act of self-regulation that has been perfected over millions of years of evolution. There is a cross-topic connection here to the world of ecology. Think of a forest after a long summer. The forest needs the winter to rest, to shed the leaves that are no longer needed, and to allow the soil to recover its nutrients. Without the winter, the spring could not be vibrant. Your sleep is your internal winter, a necessary, restorative season that allows for the spring of your waking life. It is the down cycle that makes the up cycle possible. In our time together, we have peeled back the layers of the skull to see the shimmer of the molecules beneath. We have moved past the surface of things to understand the why and the how. I hope you carry this knowledge with you, not as a set of rigid facts, but as a source of reassurance. When you find it difficult to sleep, or when you wake up feeling the heaviness of the night, remember the janitors and the gardeners. Remember that your brain is always working on your behalf, even when you aren't aware of it. It is always seeking balance. It is always striving for harmony. As you lie here now, the symphony is nearing its completion, or perhaps it is just beginning a new movement for you. The adenosine has been cleared. The melatonin signal is still strong. The GABA peacemaker is holding the curtain closed. Your synapses have been pruned, and your internal cinema has played its part. You are currently in the most restored state you will be in all day. You are chemically, physically, and emotionally renewed. There is nothing left for you to do. You have completed the curriculum of the night. You have mastered the neurochemistry of your own rest. The museum is clean, the garden is pruned, and the house is quiet. All that remains is the final surrender to the silence that precedes the morning. And so we come to the end of our time together, not an ending, really, but a place to rest. You've let these ideas settle beside you as you slept, and they'll be here whenever you return. Until then, sleep well and carry nothing but the quiet sense that you understand a little more of the world than you did before.